Hello everyone and good day. I'm Miyayang Ang, PhD student from the University of Tokyo, Japan. I'm here to present my project entitled A GWAS and Functional Regulatory Databases Approach to Identifying Key Obesity Genes. Here is an outline of my video. Often when we search about obesity, some keywords pop up immediately. For instance, body mass index, fat body mass, body weight, and others. Being overweight is extremely dangerous as it increases risk to many serious diseases such as obesity, diabetes, heart disease, hypertension, and cancers. Genome-wide association studies, or GWAS, is a major and effective method to investigate genotype-phenotype relationship by comparing DNA sequences of cases and controls, resulting in a list of SNPs significantly associated with the disease. However, this method does not provide causal mechanism. Instead, regions associated with the diseases are revealed. Therefore, making sense of GWAS results is complicated as there is no biological evidence to explain the disease causality. We cannot determine causative variants or genes by simply looking at GWAS-identified SNPs alone. A fine mapping approach is required to narrow down the list of possible causative variants and genes. One way of explaining the associations of GWAS SNPs to disease is through gene expression. GWAS SNPs could function as EQDL, influencing the expression of genes during transcription and indirectly causing phenotypic changes to a disease. This project aimed to systematically perform regulatory functional annotation of obesity-associated SNPs by implementing targeted knowledge resources. Here is an overall workflow on what we did. We first identified obesity-associated SNPs through summary statistics of publicly available GWAS datasets and calculated SNPs in LD with them. Then we performed functional annotation of the SNPs using RegulumDB and Cardio GxE catalog, revealing obesity-associated EQDL genes and GxE genes. Hereafter, we identify tissue-specific genes corresponds to gene disease association of obesity from TWAS hub and expression atlas. After assembling all the genes from the knowledge resources, we then perform protein-protein interaction analysis using string to identify obesity hub genes. And finally, we perform gene set enrichment analysis on the hub genes using Enricher. The results will be displayed hereafter. Approximately 171,000 obesity-associated SNPs were assembled, where the annotation revealed that around 89% of them falls in the intronic and intergenic region. Using RegulumDB, around 3,400 regulatory SNPs were predicted, impacting the expression of 750 EQDL genes. Some of these genes could alter adiposity and food intake behaviors in obese mice. Our Cardio GxE result disclosed that 31 SNPs were influenced by 15 environmental factors, regulating a total of 23 genes. These GxE genes include FTO, PPARG, MC4R, and TCF7L2, which often associated with monogenic obesity and type 2 diabetes. Around 550 tissue-specific genes from adipose, brain, and musculoskeletal were identified using TWAS hub and expression atlas, revealing the importance of tissue-specific gene expression during obesity progression. Integrating genes predicted from these targeted knowledge resources assemble a list of 1,224 genes highly associated with obesity. Protein-protein interaction analysis of the gene set using string disclosed 286 obesity hub genes, which involve in systemic immune repairment, insulin resistance, and glucose intolerance, implicated by studies in obese rats. Our gene set enrichment analysis revealed that obesity hub genes involved in biological processes of many cell surface receptor signaling pathways, regulating energy expenditure. These hub genes were involved in pathways of immune-related diseases, predisposing adverse consequences to diseases such as Alzheimer's disease and diabetes. On conclusion remarks, we demonstrated a workflow integrating targeted knowledge resources to elucidate functional mechanism of obesity-associated GWAS SNPs and genes. We showed that the different allele of obesity variant affects gene expression and mediating the effect of the environment on obesity through EQTL genes and GxE genes respectively. TWAS-genes and differentially expressed genes highlights the importance of tissue-specific gene expression during obesity progression from lean and obese subjects. Obesity hub genes were enriched in biological processes regulating energy intakes and significantly implicated in several immune-related signaling pathways. As a conclusion, energy expenditure could be the main driver of obesity, increasing risks for serious diseases and health conditions through high BMI. These are some important take-home messages. Computational analysis through integration of various knowledge resources could prioritize important genes involved in obesity regulation. Our study provides insights into the elements of obesity pathogenesis, improving current understanding of regulatory gene interactions and their role in obesity. I would like to thank my supervisors for supporting and supervising this project. Great infrastructures from the university and research institutes. Last but not least, the Japanese government scholarship for the fundings. Here is a list of my selected references. And thank you so much for your attention. Have a nice day.